Hi everyone, I'm Steve Burns. I'm going to be talking to you today about direct measurement of capillary blood flow in the human retinal capillary network. We made these measurements using the adaptive optics scanning laser ophthalmoscope here in the School of Optometry in Indiana. I want to acknowledge that some of the data came from Ray Warner's PhD thesis. There's Ray there. And I also want to acknowledge my other co-authors, Tom, Caitlin, and Alessandra. Thank NIH for the funding and declare my conflict of interest, which is Eon Imaging. So our goal in this study is to better understand local control of blood flow to localized regions in the retina. And what we want to get at is the extent of neurovascular coupling. How localized can we control flow in and out of a region of retina? And so to do this, we use multiply scattered light imaging of blood flow with adaptive optics. And here you can see a control subject's capillaries measured right on the edge of the foveal vascular zone, which is here. And you can see the individual red blood cells flowing through these capillaries. Now, we can't directly measure those because our frame rate on the AOSLO is about 30 hertz and we might get aliasing. So the way we deal with this is we actually make two measurements per frame. We use two imaging beams slightly displaced and we separate them out to different detectors so we get two images that are displaced on the retina at the same time. And the way to think about this is that we're constantly scanning the retina, and the scan is going to hit the same blood vessel in two different channels at two different times. So imagine these little dots are red blood cells, and in the other channel, we'll get those red blood cells a little bit later because we're further down the scan. And in our conditions, we're basically separating those two images by about 3.3 milliseconds. And so how far they move in 3.3 milliseconds is how fast they're going. And in practice, we do some image processing on the raw images. So we collect the videos, we compute a z-score for the entire video period, and automatically detect some red blood cells, and I've colored some in here. We pick a capillary and we do some detection. So here you can see several detections along this capillary, and around each detection we cut out a little region of interest, and we're going to compare the average regions of interest in one channel to the average in the other channel at this point in time. And when you do that, you get an image like this. So here's the channel we're detecting on. We see a bright spot because we're detecting red blood cells, which are bright in these images. And it's right in the center because we put the regions of interest centered on that. In the other channel, they're centered on the same location in the retina, but the blood cells are at a slight displacement due to the time offset. And so if we measure that displacement over time, we can get an estimate of the velocity. How far those cells move tells us the velocity. And here you see a series of cardiac cycles in this particular individual, and we're reliably measuring the velocity. We can do these measurements in different types of cells, but the real advantage is it lets us avoid danger of aliasing. When you get a little train of red blood cells like this, you can get a nice estimate of blood flow. Here we show a capillary going from about 0.6 millimeters per second up to almost 1.5 millimeters per second over 3 cardiac cycles. And here we show it, the measurements in a slightly larger venule in this case, and you can see the velocities higher, ranging from about 3 millimeters per second up to almost 8 millimeters per second. Now we wanted to make sure we could make reliable measurements, and so to do this, we actually broke a capillary into two segments, as you can see here, and we made measurements under different stimulus conditions with and without flicker, and what we find is we can make these measurements to roughly 2% accuracy. For the studies I'll talk about today, we used an imaging method where we put on a uniform background and we could transition it to flickering on and off, and we made measurements from a 1.3 by 1.3 degree region of the retina. And we collected measurements from venules, capillaries, and arterioles in an interconnected region. And this was so we could ask ultimately, not for this talk, but whether these all change your blood flow in lockstep. So these are the type of measurements we get, with velocities obviously showing the lowest values in capillaries, slightly higher in venules and arterioles. And what we see is we can get the same type of effect we expect from other measurements of neurovascular coupling. When we turn on flicker in all the vessels, the flow increases by about 20%. When we turn off the flicker, it starts to recover. It's not quite fully recovered here, but it's getting back to baseline, and these are not different than the baseline measurements, whereas during flicker we have clear increases in the arterioles and the venules. The capillaries are showing a similar overall increase on average, so this is the average of 9 individuals measured over time, and the main reason is this large increase in variability. I'll get into that more in a minute, but what you can see is that we plot the diameter of all these vessels versus the flow. We get a nice rational relation. It changes a little when there's flicker versus when there's not, but the data are behaving very lawfully. So we think they're quite good measurements. And so at this point, we've got a result that the data measured in arterioles, capillaries, and venules show the expected change in blood flow, but capillaries showed high levels of variability. And so that's what we want to address in the remainder of this talk. We want to look at these capillaries and see what they do when you just don't change the stimulus, just leave them steady for like 4.5 hours. How does blood flow change? And if it's changing in capillaries, and we look at, say, 3 capillaries fed by the same arteriole, are they changing the same? In other words, is it the arterial supply to a region that's varying, or are the capillaries individually changing their ability to carry the blood flow through them? And so here I show you velocities for an individual. These measurements extend over about 4.5 hours, and what you can see is the arterioles are pretty steady. We get a coefficient of variation of 5%. Another day, we get a coefficient of variation of 6%. Venules, we get coefficient of variation of about 9%. But the capillaries are varying a lot, 18 to 23% in their blood flow. And this is basically what we saw before, but now we're showing it over long time periods without changes in visual stimuli. This summarizes all the participants. There were 3 participants and multiple measurements. And you can see arterioles and venules have a coefficient of variation in their velocities on the order of 5 or 6%, whereas the capillaries are varying over about 20%. And this is not stasis. So one of the things people ask is the blood flow just pausing? So here I'm showing you a high velocity and a low velocity for this particular capillary. And what you can see is the blood is flowing in both cases. It's just the overall flow is varying. And we can see stasis, but it looks more like this. And so we can see that here a rouleau has gotten lodged into the capillary and it's blocking flow for more than a second, whereas here we're seeing no stoppage of flow. So the second part of this experiment was to ask, does capillary variability reflect changes in the supply from the arteriole? So we picked 3 capillaries fed by the same arteriole and measured how they changed their flow. And the answer is they changed separately. So here's just an example. Here's at time 29 through 33 seconds, 3 capillaries, different colors. And you can see that 100 seconds later, we actually made many. This is just an example, but now the one colored in red has much higher flow than it did earlier, whereas the one colored in green has lower velocity, and the one in purple is a little bit lower velocity. And when you look at multiple measurements under different conditions, you can see that these capillaries are changing differentially over time. That is, they are varying independently. In this capillary 1, during the no flicker period, it actually had high flow, and in this one, it had low flow. So the flow increased and the flow decreased. And this is, again, what we saw with our group data, but now we see it in individual capillaries fed by the same arteriole. So our conclusion is that neurovascular coupling can be reliably measured down to the smallest arterioles and venules. This opens a possibility of looking at the size of the neurovascular unit and understanding how local control is in the retina. But at the level of the capillaries, there's other processes going on that seem to be involved in control of blood flow. And so while on average they do show the impact of neurovascular coupling, there's an additional source of variability where the capillaries seem to be switching through various levels of flow independent of the local demand as applied by us. That may be responding to local demand in terms of local signaling from the retina, but it's at a local level. And so that's our talk. Thank you, and thank you for your attention.